Good morning, everybody. We start the second session of this final conference, which is implementing FAANG innovation for precision breeding. I will give a first presentation that is an introduction to the first part of this session. As yesterday, finally we think it's better probably that we try to answer as soon as possible to the questions that we get in chat. So please add your questions to the chat, and the speakers will make their best to reply as soon as they can. Okay, so I start with my introduction. It's not the first slide. Can you put my slide? Okay. Okay, well. So the title of this part is Influence of Maternal Diet on the Epigenetic Programming of Offspring. And it is a part of a project co-led by Wageningen and INRAE, by Jerry Wells and myself. This one, okay, sorry. Okay. So in the concept of gene switch, well, this figure, let's say, shows what is the concept of the whole gene switch project. So in the upper part, you see functional annotation across tissues and developmental stages. This was the subject tackled yesterday by the different presentations. And below, you see how we use functional annotations for precision animal breeding. And this is made of two distinct parts. On the left, the one pertaining genomic selection research that will follow after the next presentation. And this one, which was thought to, in a broad perspective, that is, can we find a complement to genomic selection practices like using lower quality, more sustainable feed, and perhaps that could also provide better protection from disease. And the background concerns the microbiota, because the key metabolic activity of a colon microbiota is the fermentation of non-digestible carbohydrates that lead to a variety of microbial metabolites in circulation. And propionate, butyrate, and acetate are major end products, short-chain fatty acids, or SCFAs. And these products have become very much relevant in research in the recent years. Here you see in the picture just the snapshot of their main path of synthesis that you have for these metabolites, not synthesis but production. And these provide, first of all, a quite substantial contribution to the caloric requirements of humans and mammals in general. So it's thought to be about 10% for both humans and pigs, and much more for the ruminants. And we are considered, these molecules, as major players of intestinal homeostasis via G-protein-coupled receptor, dependent signalings, and also epigenetic mechanisms. And there are a number of effects that have been documented in the gut, in these roles in homeostasis, like ability to increase regulatory T cells to control inflammation, induction of digestive hormone production, and in general, the induction of gut defense molecules. And the SCFAs have effects on tissues and organs that are not only the gut, but also beyond the gut. Because these molecules are transported, they are absorbed by the gut epithelium, and so they enter the circulation, part of them, some more than others, but they enter the circulation via the portal vein. And in time, there have been evidences that there have been effects that can be documented at different tissues, including regulation of energy metabolism signaling via GPR signaling in sympathetic neuronal ganglia, for example, so a regulation in the neuroimmune axis. The acetate increase of Tregs and immune regulation via FOXP3 acetylation, and so on. So you see examples here. And including also in the bottom the epigenetic imprinting of bone marrow hematopoietic stem cells. And you find several reviews on these effects that are documented, more or less sure, more or less robust, of course. But what is interesting is that indeed, after clearing in the liver, especially acetate can reach quite high concentrations in circulation, in the venom serum, in both human and pigs. And so probably this is one of the reasons why we can really detect some effects, including epigenetic effects, that are triggered by this molecule. I also put an example of one of the many papers where in this case, propionate, sorry, but seems to regulate megakaryopoiesis, a megakaryopoiesis that produce platelets, and so an affection on the function of platelets triggered by propionate. And at the time that we thought of this specific part of the project, and it was quite some time ago now, because we started to think of this in 2017, we had found a very inspiring paper, that is this one, Gut microbiota alter host histone acetylation and methylation in different adult tissue of mice, and mainly by short-chain fatty acids. And in this paper, we found that gut microbiota can alter host histone acetylation and methylation in multiple tissues using germ-free mice, conventional mice, and recolonized mice for the microbiota. They demonstrated that Western diets, similar to Western diet, so diets that are poor in fibers, can suppress the microbiota-driven SCFA production and chromatin effects. And the supplementation of germ-free mice with SCFAs alone was sufficient to recapitulate chromatin modification states and transcriptional responses associated with gut colonization of a microbiota. So it seemed really from this paper that SCFAs were the key to understand this effect of a microbiota on the, also on the epigenetic imprinting of the peripheral tissues of the gut. And the other component that we considered was that epigenetic imprinting likely occurs in utero. Now, in human research, this is a very important debate about how fetal life can affect health, also the adult, also in the later age of the offspring. And of course, the same mechanism, you can think that they can affect the fetus. And so the fetus could have some modifications that are maintained and that you can find again also in the offspring. And at the time, we had found, among others, one paper where they demonstrated that asthma is of developmental origin, and it's influenced by maternal diet and bacterial metabolites. And this was another important communication that was quite inspiring for this work. And in the animal nutrition field, this is also, and has become even more in the recent years, a very important topic. Back in 2012, they found that butyrate supplementation to gestating sows, and this was really with butyrate given in the water in very high concentration during the late part of a pregnancy, was actually triggering an effect. So piglets were born with higher body weight. And more recently, there have been studies more really addressed to use fibers in specific concentration in the feed. And I put two of them. One is the second one, methyl donor nutrient, micronutrient given to gestating sows. and they showed an effect of gut microbiota metabolome in the offspring. So they thought, for example, they found the increase of microbiota diversity, uniformity in offspring piglets, SCFase concentrations, and metabolism of 21 days piglets. So in general, a beneficial effect of this treatment. And in the lower one, again, they claim that this treatment with diets improves performance of the sows and reduces inflammation in sow and piglets. And this is quite interesting also for the research that we present later. And actually, they didn't find really significant differences in piglet performances at the level of weight. But in fact, they could observe lower levels of inflammatory biomarkers, both in sows and in piglets. And this, as you will see, is going quite nicely together with the results we obtained in our study. So what aims that we had here? The aim is to assess the epigenetic effects of maternal diets, varying for fiber contents on pig fetuses and post-twin piglets. And what we thought as hypothesis, first that epigenetic effects, assuming they're triggered mainly by SCFAs, occur to the pig fetus via the placenta and exchange mechanism, so it's something that happens during pregnancy. But the epigenetic effects could be detected in the pig offspring up to at least 10 weeks postpartum. That means that after the generation of piglets was born, they were kept all under a same standardized diets, far from the mother, and were sampled 10 weeks of age. And these effects these maternal effects can be detected in the genome at the transcriptional and chromatin accessibility levels. So we performed the bulk RNA-seq and ATAC-seq. And we targeted liver and skeletal muscle. Why? Well, first, and there have been also, as you see on the right, increasing evidence about this on the gut muscle crosstalk, etc. There are several pathways related to metabolism that can be affected by SCFAs, and particularly by acetate, which should be always the most abundant in circulation, like upregulation of fatty acid oxidations in liver and muscle, and also protein metabolism in muscle. And both tissues are central tissues in gene switch, because also from the work that we presented yesterday, for these tissues, we have the most complete functional annotations. And in the case of a fetus, we characterized, annotated quite carefully the fetus at the same age of 70 days post-fertilization. And in addition, and this will follow in the second part of the project, a EQTL study has been performed on several animals, and liver and muscle were target tissues as well. So this was a bit our strategy, and as I said, since recently, also especially for the muscle, this is an example I report here. There are nice reviews where you can find more about the gut muscle crosstalk and about what they find of the influence of intestinal microbiota on growth and physiological state of the muscle in pigs. And another thing is about the choice of the epigenetic assay, but in our case was ATAC-seq. And this, well, you know, very often people investigate this kind of mechanism by using methylation profiles. In this case, we made a different choice based on evidence, because from the same paper by Kraut-Kramer et al. 2016 that I showed before, there they made a quite nice biochemical assay, actually, by mass spectrometry, and they did it in different tissues, in the different cohorts of mice that were differently treated. And they found that they had quite an effect on the global histone acetylation and methylation, and that you can find it. And seen on the right, ATAC-seq is not a specific assay, but in general, as from the original work of Buenrostro, that is the discoverer of ATAC-seq, he described it 10 years ago. Usually, ATAC-seq is quite correlated to ChIP-seq marks of histones. So we thought it was a good compromise to avoid we could not do it anyway, even if we wanted, to make very different assays for acetylation, etc., that we could not afford. So we thought it was a good compromise. And the experimental design is this one. So we had a total 21 sows from similar parity. And to these sows that were distributed in 3 groups, we provided diets that were either controls, so low fiber, or 2 diets based on beet pulp fibers or pea internal fibers that were administered during essentially most of the pregnancy from day 2 to day 108. And each of these sows had 2 subsequent pregnancies. In the first one, piglets were produced, and from each one we sampled 2 males and 2 female piglets. In the second pregnancy, we collected the fetuses, so also the sows were sacrificed with the same skin. And these sows, I also insist on the fact that we made this large experiment, in fact, in 2020. It was COVID time, so it was a kind of a challenge to carry it out. And they did this in Wageningen at SFR. This was our subcontractor. And finally, it was successful. And the other thing is that we worked really close to commercial conditions, I would say. Because, for example, you see the genetic lines. This is not the single genetic line for all the experiment. We used different sows from 2 lines of topics, Norsving genetics, and also different bores. And about what measures we did on this experiment, of course, the SFA measurements and analysis from blood serum and feces, depending on sows and piglets at the time, and also microbiota characterization. And for fetuses and piglets, we collected the weights, including position in uterus for the fetuses, SFA measurements, and of course, we profiled transcriptome and chromatin accessibility. And I stop here because I want to give the floor to Andrea. And this is the slide that we will show again also after her talk. So the acknowledgements of all the people that have collaborated to this work. I will not go into details, otherwise it will take too long time, and I already taken quite a lot. So thank you very much, and I give the floor to Andrea Rau.
Good morning. I'm Andrea Rau. I'm a researcher at INRAE in France. And I'm pleased to, we'll follow up this nice context provided by Elisabetta for this part of the work that we carried out in GeneSwitch. I'm speaking to you today as a statistician disguised as a biologist. I helped to coordinate a portion of the analyses that were performed within this complex set of data that were generated for this project in collaboration, notably with Sman Sharabi, Sarah Jabadi, and Linda Lunin and Jose Bokhorst. So to start off, we'll ease into the presentation with the same experimental design that Elisabetta just showed us. So as she mentioned, this ended up being a relatively complex experimental design, notably due to several factors. The primary question of interest here, obviously, was looking at these effects of the maternal diet on the offspring of these sows. And several measures were taken at the sow level, at the fetus level, and at the piglet level. We also made quite a bit of time at the start of this project as we were defining experimental design, trying to optimize the power calculations. So that's something I'm skipping over entirely here, but keeping some guesses in mind of what we expected to see as in terms of effects. We tried to determine what were reasonable sample sizes, how to best control for litter effects, and so on. And so here we're really trying to balance being able to have enough animals available to detect what we anticipated to be slightly subtle changes, but also balancing the animal welfare and budget considerations. So as Elisabetta said, the experimental design was already fairly complex when we set it up in our ideal optimized framework. And then, of course, there was the reality of the fact that this experiment was carried out in these indeed pseudo-commercial conditions, we can say. So the fact that we had a couple of additional sources of variability that needed to be accounted for in the model, including the fact that we had these two different breeds of sows and two different breeds of boars that were not originally intended in the experimental design. There are also some additional complications in terms of some of the interpretation of results. So for example, the diets were administered to the sows 2 days after insemination. There's a little bit of wiggle room there, and in terms of the timing of certain measures, but I'll come back to that in a moment. So to start out with, we had this huge set of data that we're looking at these different levels of the sows to fetuses to piglets. And so the first thing that we wanted to evaluate was the extent to which these different diets, so low fiber and our two high fiber diets, impacted the sows at a phenotypic level. So we can take a look here at a first analysis which shows us a projection of the sows we considered in the experiment. So we're looking at fecal 16S microbiota data for the sows. And what you'll notice is that as the points here are colored in our first two principal components from an RDA analysis, according to diet, we do see that there's a clear separation among the three groups of sows. So the dietary intervention is indeed impacting the sow microbiota as expected. And we'll see also that because the sow microbiota was measured at day 0 and then following up day 30 to 70 in the first pregnancy and 70 in the second pregnancy, it's post-intervention that we see these differences. So all this lines up with expectations. However, if we are looking up to whether our expectations are met, when we look at the short-chain fatty acids in either sow serum or sow feces, here we don't see a clear impact, particularly with the SARA measures. So the plot here on the left is showing along the x-axis the different time points throughout the experiment. And on the y-axis we have, I believe, the acetate concentrations in the sera in the left-hand plot, and in the fecal samples in the right. So again, we see our same three colors we'll see throughout the presentation representing the three dietary groups. And clearly there's not enough signal present here to determine a clear distinction between the dietary groups. We did statistical tests here to evaluate whether there are any differences. There's nothing significant. But again, in terms of the complexity of the experimental design and how the experiment had to be carried out, I should note that there is some heterogeneity that was present relative to the timing, notably of the serum samples after feed intake of the sows, which obviously can have an impact on those acetate levels. So this is maybe more inconclusive than a negative result. Okay, so we've seen that these different maternal diets do seem to be impacting the mothers, but then we can also look at phenotypically whether those different diets appear to be impacting the offspring. So whether at the fetus level or at the piglet level, accounting for different factors such as the mother-father breed effect, the position of the fetuses in utero, eventually litter effects, so these are on the fetus side, or for parity, litter size, et cetera, for the piglet size. We tried to determine whether these different maternal diets actually impact the weights of the offspring. And here we see that, again, there does not appear to be an impact of these different maternal diets on the offspring weights themselves, so the size of the fetuses and piglets. Okay, but then we can take it down to another level and try to determine whether these maternal diets are impacting the piglet SCFA levels or the piglet microbiota. And here we see sort of a partial impact. On the left-hand side, we're looking at the piglet fecal samples to determine different SCFA levels. We have 3 different SCFAs on the x-axis. And what we see is essentially some stars, right? There's some significant effects that are appearing between diets, but far from being all of the different short-chain fatty acids that are considered. On the right-hand side, what we see is, again, at the piglet level, a similar plot to what we saw from the sows. So notably, we're looking at a redundancy analysis to the right. Where we're looking at essentially it's a supervised PCA. Is it my phone? No, something over here. It's not my phone. There's just something beeping up in the corner in the wires up there. So on the right-hand side of this plot, we see again this supervised PCA analysis called a redundancy analysis, looking to separate the 3 maternal groups of the piglets this time rather than the sows. And what you'll notice is compared to the previous plot, although there's a little bit of separation between the maternal groups, it's far from being as marked as we saw for the sows. And in fact, when you run permutation tests to evaluate the significance of this separation, and once we account for litter effects of these piglets, that's where we see that there's not strong enough impact to be detected statistically. Okay, so that means that we've evaluated sort of at a phenotypic level what these impacts of the maternal diets are, and then we can get into the heart of the question that we looked at, which was the impact on fetal and piglet chromatin accessibility and gene expression. So we already saw a bit about the different pipelines that were defined within GeneSwitch to analyze both ATAC-seq and RNA-seq data. So here on the ATAC-seq data side, well, the data are now publicly available. The NF-core ATAC-seq pipeline was used to analyze the data. And when we get into questions of differential analysis, of the differential chromatin accessibility, we made use of normalization procedures that are adapted for these data. On the RNA-seq side, similarly, the Tagada pipeline that Sarah and Sylvain presented yesterday was used to analyze these data. Because the characteristics of RNA-seq data are somewhat different from ATAC-seq data, it means there was a different normalization procedure used within the differential analysis pipeline. And similarly to the ATAC-seq data, these data are publicly available with the accession numbers that are recorded here. So beyond this question of appropriate normalization methods, for each of these 2 data types, there was a similar model that was used for the differential analysis. That is the model which is looking to identify peaks in the chromatin accessibility data or genes in the RNA-seq data that are showing significant differences between the maternal diets, controlling for all these other experimental factors I've already mentioned. So we had covariates corresponding to sex, the breed of the mother, the breed of the father, and a random effect to account for correlations within litters. Okay, so a typical first step when you're looking at these high-dimensional omics assays is, of course, to do some dimension reduction and visualize the samples on, say, the first 2 components of a principal components analysis. So what we see here to the left is the principal component, first 1 and 2, for the ATAC-seq data, and on the right for the RNA-seq data. Points here each correspond to a single animal, so fetus or piglet, and the points are colored according to the maternal diet. So we have the same colors of gray, blue, and red. And essentially, what we see here is that there is a strong separation between the tissues on the first principal component, and between the two developmental stages on the second principal component. So a fairly unsurprising result. We see these in other omics data assays that such strong separation exists between tissues and between stages. But what this means is that essentially for both ATAC-seq and RNA-seq data, the choice was made to focus on combinations of stage and tissue and perform differential analyses within each of the four combinations. So if we take this is an example of one of the ATAC-seq data sets, if we focus on the combination of piglet muscle, so again, same type of representation where we have PC1 versus PC2. Each sample represents a piglet here, colored by maternal diets. And if you're looking for separation among the maternal diets, you'll likely not find it. So there's no strong global overarching genome-wide distinctions between the maternal diets. And we see similar figures when we look at other combinations of stage and tissue and for the RNA-seq data. So usually, when I'm running a differential analysis, that's typically a sign of things to come, which is to say that there won't be huge numbers of differential signal, so peaks or genes, and the effects that we will be able to determine will be somewhat subtle. Okay, so but before we get into the results of the differential analysis itself, we also wanted to take a look at the extent to which our ATAC-seq and RNA-seq data appear to be sharing signal, so co-varying together. And one way to go about doing this is looking at what's called a co-inertia analysis. There's a sort of schematic illustration on the bottom of this slide, which illustrates essentially when we have two data matrices, ATAC-seq and RNA-seq, for a given stage and tissue. We can calculate what's called an RV coefficient, which is essentially a multivariate generalization of a standard correlation. That gives us a number to quantify to what extent the two tables co-vary together. And we can calculate this number for each combination of stage and tissue. And then we can actually evaluate to what extent this signal is strong by permuting the rows of one of our matrices. So you mess up the covariation between the matrices, and you do that a whole bunch of times. And then you look at whether your RV coefficient from your real data is a lot larger than what you would have seen if you mixed everything up. And in that case, you'll have a small p-value, which shows that the signal you're detecting does appear to be stronger than what you would see randomly. So what we see here is this covariation signal, this RV coefficient, appears to be strong enough for detection with this permutation test in all the four data sets, with the exception of the fourth combination, so it's the muscle fetus, where the permutation test was not significant in this case. Okay, so using a similar type of analysis, we can also look at the extent to which there's shared signal at the piglet level between the transcriptome and ATAC-seq data on the one hand, and with the microbiota 16S data. And what's quite interesting here is that we see that, in fact, the RNA-seq data do appear to be significantly co-varying with the microbiota data, but that's not the case for the ATAC-seq data, which here are insignificant after those permutation tests. Okay, so now we'll finally get into the results from these differential analyses, trying to assess where we're seeing different expression or different chromatin accessibility between our maternal diets. These tables look awfully small on these screens, but maybe it's big enough for you to see that the numbers are quite low here when we're looking at the two tissues, so liver on the top followed by muscle. We've split signals into up expression and down for the various comparisons. And each column here provides a comparison between pairs of maternal diets. So low fiber P versus, high fiber P versus low fiber to the left, high fiber beet versus low fiber in the middle, and then the high fiber beet versus P on the right. So what you'll see is, yeah, we've got, this is ATAC-seq data, so we're starting with 200,000 some peaks, and we're looking at 0, 1, 2 differential peaks at the fetal level. We see a little bit more happening when we move to the piglet level, which is to say, particularly for piglet muscle, now we're starting to see a bit more signal about 650 some peaks that are differentially accessible between the high fiber beet and low fiber diet. Just for reference, I'm also including some of the numbers of differential peaks for these other covariates that we included in the model, notably sex, the breed of the mother, the breed of the father, and that's it. So you'll see that, in fact, these covariates in our experimental design are carrying more signal than what we're seeing in the differential analysis of these maternal diets. We did check whether there was any interaction between the sex and these maternal diets in the ATAC-seq data, and in fact, perhaps unsurprisingly, there were no interaction effects found, which could be a power issue or not. And I should also note that we checked whether there was any, here I'm talking about differential expression that was found for mitochondrial genes. What I should say is that a similar table for the RNA-seq data was essentially zeroed out everywhere for the maternal diets. So there were no differentially expressed genes that were identified between maternal diets. Okay, so commonly a thing we do with these ATAC-seq data is we try to look at how the peaks that were identified as being differential are situated within the genome. So what we have here is just a pie chart on the left of the full set of peaks. So we've got 240-some thousand peaks in total, according to their different genome annotations, exonic, intergenic, intronic, and so on. And this distribution of peaks is fairly standard. It's what we see in other experiments as well. And when we compare this distribution of genomic annotation for differential peaks compared to the all peaks, what we see is essentially a bit of an enrichment in intronic and promoter TSS peaks, which again is somewhat aligned with what we've seen in other experiments, so that these peaks in these particular regions would tend to be more actively involved in these biological processes. Okay, so taking things a little bit further beyond these differential analyses, the idea was that we can actually go beyond using these ATAC-seq data to ask some fairly interesting questions, notably about transcription factor footprinting. So TF footprinting is in fact what's illustrated by the figure on the right. What we're essentially looking for is peaks where we've got this footprint, this dip in the middle, which essentially corresponds to a portion of the genome where there was accessible chromatin, but a transcription factor has come to bind to the middle of that open chromatin, and so the peak dips at that spot. So not only can we tell that the chromatin was open, but we can say something about to what extent that open chromatin was actually allowing for transcription factor activity. So how do you go about doing this TF footprinting in practice? Essentially, there's a first step where you predict where the footprints occur. So this is happening from all 240,000 peaks that we've detected with our ATAC-seq data. Then, once you've identified these portions of the genome where you've got these dips occurring within ATAC-seq peaks, then you can go back and match up the transcription factor binding motifs that you can pick up in various databases, for example, Jasper. And you can try to match up those footprint spots with these transcription factor binding motifs. And then what happens is we can go to our data themselves and actually try to look at the average chromatin accessibility around each predicted transcription factor binding site, which then essentially allows us to look at a measure of the extent to which there appears to be differential transcription factor activity between our maternal diets using a tool here that's called a hint attack. So yeah, this is all, it's pretty cool because we're only using the ATAC-seq data at this point. This is not at all touching the expression data itself. Okay, so what's interesting is we're thus able to pick up on these tissue-specific differential transcription factor activity results. And we're illustrating them here with a figure. It's a heat map where we've got on the columns of the heat map are corresponding to different comparisons of maternal groups. In the liver are the first three columns, muscle are the second three. And what we're seeing here in the rows of this heat map are essentially transcription factors that have been determined to have these differential activities between maternal diets. And we've got the red to blue scale to indicate whether it's going up or down in each comparison. So a total of 21 transcription factors were identified in the fetal analysis, 30 in the piglet analysis. And what's quite interesting here is mostly what we're picking up on is the fact that because these heat maps are divided into tissues, according to the first 3 and second 3 rows, we're seeing a lot of tissue-specific activity. So we're going from red to blue or from blue to red when you compare the 2 halves of the heat maps. We can also pick up on, I've highlighted here just one particular example that caught our eye, which was that the NFY transcription factor has been picked up both for piglets and for fetuses. And this is a particular transcription factor of interest as it's been linked to stem cell fate decision and regeneration in adult skeletal muscle. Okay, so as I mentioned before, we didn't have any differential genes when we performed that differential analysis from the RNA-seq data. So one thing that is often done in cases such as this is to move from focusing on those individual gene level expressions to looking instead at gene set expression. So what we're essentially looking at is, rather than saying for each gene, is it going up or down for whatever comparison, we're saying for a given set, are we seeing coordinated expression changes that maybe are too subtle to pick up individually, but collectively have enough power to be detected? And so that's what we're going to look at here in this objective to sort of boost the power of signal detection from RNA-seq data. So to do this analysis, we made use of a tool called Ermine-J. And we are going to focus on the significantly altered biological processes that we identified in piglets and in fetuses in our 2 high-fiber diets versus the low-fiber diets. So if you've ever run a gene set enrichment analysis, you get results like this, a huge table where you have gene set names associated with a p-value, that are ordered according to p-value or set size. But then the idea is to try to figure out how can you say something about this giant table of results. There were a certain number of pathways that were flagged by this analysis. And one way to do this is it's a fairly creative visualization that Jos Bokhorst came up with when looking at these results. So we'll focus here on the result where the high-fiber beet diet is overexpressed compared to the low-fiber diet in piglets. And what he was able to do is to propose this sort of graph-oriented representation of that table of gene sets I showed a moment ago, where we can group gene sets according to related functions. And unfortunately, the screen's a bit small to fully appreciate it, but this graph is extremely rich in presenting whether this overexpression is present in muscle alone, in liver alone, or in both. So notably, each node in the graph represents one gene set. Gene sets that are sort of lighter on the outside are only present in liver, darker on the outside only in muscle, and if it's lit up red throughout, it's in both samples. So we have a lot of tissue-specific processes in this particular graphic, and redder nodes correspond to those that have higher expression in the HFB diet compared to the low fiber. So what we're seeing here is, in particular, what caught our eye was the gene sets towards the right of this graphic. We've got a giant group of metabolic processes and carboxylic acid transport. I always say that one wrong. And here we're looking at gene sets that were primarily expressed in liver. So this seems to be going in line with what we could expect. Okay, so then we can also look at the opposite. If we're looking at genes that appear to be more weakly, gene sets that appear to be more weakly expressed in high-fiber beet compared to low-fiber diets, we'll see a similar picture where we're grouping gene sets together. But here, the picture itself looks quite different. We've got a lot more of these bright red nodes. So we have shared underexpression in high fiber diet compared to low fiber between the two diets. And here, the groupings of gene sets are really highlighting gene sets that are related to immunity. So immune response to immune cell activation and differentiation in both tissues. So it's a quite striking difference when we're looking at underexpression in these high fiber beet diets compared to the overexpression on the previous slide. Okay, so the sort of one way to look at trying to wrap up this whole picture of these analyses that we did is to understand, like, if we take this example of the piglet underexpression in high fiber beet compared to low fiber, we saw immunity lighting up everywhere, and we were wondering whether we can somehow make the link between these immune-related pathways to actually looking at immunity cell types. Or if I rephrase it in particular, is the overexpression of immune-related pathways in these low-fiber diets due to an overabundance of immunity cell types, or to an overexpression of the genes in those gene sets? So we have some ongoing work on this particular point where we were looking at making use of deconvolution methods from the pig atlas, the pig GTEC atlas, right? That is making use of bulk RNA-seq data to try to benefit from cell type-specific markers to tease out which specific cell types are present in our bulk data. So doing this deconvolution on a specific, so we use the deconvolution on our full RNA-seq data. And then what I'm going to look at here is just focusing on genes that are both in those immunity pathways I highlighted a moment ago, and genes that are tissue-specific marker genes that have been identified for the deconvolution analysis. So what we see in this heat map here, on the columns of the heat map correspond to different cell types. The rows correspond to these tissue-specific marker genes that we're focusing on for our comparison. And our first plot here corresponds to piglet versus muscle. So what are we looking at here is that we see that essentially the left-hand portion of this heat map is all the immunity cell types. So we have notably mature B cell, macrophage, NK cell, et cetera, which are the genes that are really lighting up in the liver samples. When we do the same analysis on the liver side, so we're still focusing on these immunity-related genes in our immunity-related pathways, what we're seeing here is it's not as lit up, right? There's not as much yellow in this heat map, and so the average overall expression in our samples appears to be lower in in muscle compared to liver. So all this seems to be suggesting that through deconvolution, we're able to determine that there are pertinent biological processes corresponding to pertinent cell types in these samples. So with that, I'm going to arrive to the sort of a summary of the overall impact of these maternal diet interventions. So you'll notice that I focused on 3 different levels of analyses. The first, in terms of phenotypic measures, showed that after these dietary interventions, the sow microbiota does indeed cluster by diet, as expected. And we see some trends in fecal SCFA content, although those did not reach statistical significance. For the offspring, we note that the piglet fecal microbiota and SCFA content appear to be explained, but only in part, by those maternal diets, so an impact much less strong than what's observed by the sow effect. At the next level, then we looked at chromatin accessibility data. So we did see that there was some significantly differential accessible chromatin between those maternal diets, in particular for the piglet muscle combination. And what's interesting is that we see that those high fiber diets in the mothers does appear to trigger clusters of transcription factors that have very tissue-specific effects. And finally, in terms of transcriptomics, we see that there do appear to be these effects of high-fiber maternal diets, but at the gene set level and not at the individual gene level. So these changes are primarily associated with immunity and metabolism processes. And finally, the deconvolution analysis of the expressed genes from adult pigs in a single-cell transcriptome atlas does suggest that these proportions of immunity cell types in the tissues may be triggered by different maternal diets. So I'll just wrap up with 3 quick take-home messages. The first is that indeed the effects of maternal diets differing in these fiber contents does appear to impact animal phenotypes, whether the SCFAs or microbiota, does not appear to affect animal phenotypes. As we saw, the offspring weights were not affected, so we're either missing statistical significance or those effects just aren't there. Both transcriptomic and epigenetic effects do occur in fetuses, but where we saw the most signal appeared to be in piglets. And this is piglets that are 10 weeks postpartum. And as Elisabetta said, I just want to stress those are piglets that were all raised in the same conventional diet, that were separated from the mothers quite early on. So the only differences that we should be seeing between those piglets are in fact what their mothers were given as food. And finally, these high fiber sow diets did appear to lead to enriched biological pathways in piglets, and that's consistent with the physiological changes that we observed, and notably those that are associated with immunity and metabolism. So we have this idea that overexpression of immune-related processes in low-fiber diets could be suggestive of the role that's played by these SCFAs with respect to epigenetic imprinting. So with that, I'll end on the same acknowledgement slide that Elisabetta showed earlier. There were lots of people that were involved in this work, and I do want to highlight in particular those who contributed significantly to the analysis effects. So Wageningen, Linda Lunin, Jos Bokhorst, and Jerry, who was part of the coordinating team. At INSERM, Sarah Jabali, and at INRAE, Sman Shalabi, Elisabetta, myself, who worked with these data throughout.
Okay. Good morning. Welcome with the part 2 of this section on implementation of FAANG innovation for innovation breeding. As mentioned, my name is Marco Bink. I'm from Hendrix Genetics, and together with Mario Calus, I was leading work package 4. And I think you've seen this one before. So this is the diagram of the gene switch organization. So we are part of Pillar 2, and Pillar 2 also contains the epigenetic work that was just presented by Elisabetta and by Andrea in work package 5, and work package 4 is the part on genomic prediction or building genomic prediction models. A quick reminder about genomic prediction. Very simplistic. So what is the rationale is that we have a population of animals, a reference population, where we collect tissue or blood samples, and we also collect trait phenotypes. From the tissue or blood samples, we extract DNA and we perform SNP genotyping, quite often about 50,000 along the whole genome. And with this information, we built a genomic prediction model. So we tried to make a model where we calculate for every SNP how important it is to predict the phenotype. Once we have this genomic prediction model, we can apply that to a validation population where we do collect the blood samples or tissue samples, and together with the prediction model that we have established from the reference population, we can predict the breeding values for a certain trait, for example, for the daily growth. And then here for this simple example, you see the breeding values of these 4 piglets. And as a breeder, you want to select only a few of them to breed next generation. And you would select then those that have the highest breeding value. So with that, you have this prediction model, and that has been proven to be very successful, has been implemented by many breeding companies in all livestock species. And the assumption is that every SNP is equally important in this equation. So we assume that every SNP explains about 1 over the number of SNPs times the genetic variance. And then the idea is to within GeneSwitch, but also in other initiatives, other projects. What if we can relax this assumption about every SNP being equally important by utilizing functional information as these are emerging from FAANG projects like GeneSwitch? And here it says that we use that as a prior, but you can do that in different ways, as we'll see later on. One step back is to consider the genome to phenome gap, and we in the genome prediction model, it's quite simple. We go from the DNA level to the trait level, so from DNA to, for example, feed intake. But as we have heard before yesterday and today, we generate more and more knowledge about the genome structure, genome accessibility, etc., with all kind of assay techniques. And we can try to utilize that information about where are the genes, whether they are accessible or not, etc. And there are two ways to capture that intermediate information. So either by performing it on an individual level, so we're going to measure gene expression for our animals, so we're going to measure methylation for animals, or we can do it on a population level and take that functional information from that path to into the genomic prediction models. So if you think about the breeder's perspective on estimating estimating breeding values, the main focus is on improving prediction accuracy. So what we have seen before is that from the standard until 10 years ago was we used pedigree and phenotypes to calculate breeding values. And then with the arrival of SNP arrays, affordable commercial SNP arrays, we could genotype all our animals and we would add the genotype data into the prediction model. So and we did see about an increase, depending on the trait of interest, between 10 and 50% increase of accuracy. And then, of course, the question is whether, then one step more. So we saw an increase by using SNP array chips with 50K, and then, of course, we did try to say, okay, what if we can infer and can use all SNPs along the genome, so imputation to whole genome sequence level. People have done that, and simulations, it worked quite well, but in reality, we only saw a very marginal increase from 0 to 5%, and it was more to 0% than to 5%. So that didn't bring a lot of gain yet. Then the question is that, okay, what if we add annotations? How much can we expect of the increase in prediction accuracy? And then, so we have all kind of different maps, and we can try to build new prediction models utilizing every map individually. And we have tried that within GeneSwitch as well, and I think there are some results on that in the next presentations on the methylation scores. So that's one approach, but that is less flexible, I think, and quite cumbersome. The other approach could be that you try to think about a kind of scoring mechanism that integrates all these annotation maps into a single score, and you use that score to be added into the genomic prediction model. So examples of that are CAT scores or FAT scores. And then the main difference between CAT scores and FAT scores, in CAT scores, you do not take the trait of interest, phenotype traits of interest into account. So it's a very bioinformatic based approach. You do use a lot of information about evolutionary mechanisms, etc. But in the FAT scores, you train the model using phenotypes that you have collected on your populations. So that's as a kind of introduction. Then about the presentations in this session. So in total there are 5. This is the schematic overview of the work package 4. Different tasks. There are 6 tasks. And then the first presentation by Mario will focus on model development. So we have worked on Bayesian approaches and machine learning approaches. The Bayesian approach was mostly done by INRAE, by Andrea and Fanny Molandin. And the work on the machine learning was mostly done by Bruno, but Mario will elaborate on that in more detail. Then there are 2 presentations about identification of QTLs. Maria will, Ballester will focus on gene expression analysis, genes expression data collected on 3 different populations, each population about 100 animals, and she'll allude further on that. And then the other presentation by Carl-Johan Rubin, he is online, and he will present about fine mapping QTLs in chickens. And then the last 2 presentations are by Bruno Perez and Andreas Kranis, and they will focus on these FAT scores that I mentioned, and also Andreas will focus on how effective or how could we utilize functional information to subset SNPs, so in that way to improve genomic predictions. Again, so here the list of speakers, where I made a mistake that the presentation by Bruno, it will be before the one of Carl-Johan. So we'll have Mario about model developments. Then we have 2 presentations in pigs, and then 2 presentations in applied to chickens, broilers. That's it. Any questions? If not, then I think we can continue with the first speaker, Mario.
So as Marco also briefly mentioned, what we were concerned with in work package 4 is that, well, what we typically do in genomic prediction, we have the genotypes of the individual, and from that we make sort of a linear relationship to the phenotypes observed. But we know that in reality, biology is not so linear. So the idea was, if we know more about this genotype to phenome gap, can we use that information in our models to make them better? And so just to quickly take you through the overview of work package 4 again, typically in genomic prediction, the applications as they are today, the inputs are genotypes and phenotypes of individuals, perhaps also pedigree, but genotypes and phenotypes is the main source that we put in our genomic prediction models. The idea was that in work package 4, we would extend those models to be able to use functional annotation information generated in work package 1 and 2 of a gene switch, but possibly also collected from elsewhere. And next to that, we would have both FineMap QTL and eQTL that were generated within work package 4. And then finally, those models were evaluated in validation in commercial-sized population, as also will be presented later in this session. So first of all, let's start about the Bayesian genomic prediction models. So the idea is that with those Bayesian models, they are useful for 2 things. One is for prediction of breeding values, but also because of their ability to differentiate between SNPs, the effects between SNPs, and also the variants associated to it. You can use them for QTL mapping purposes as well. So the idea was that if we can bring functional annotation information into those models, we may be able to enhance both of these functions of these models. So the starting point was the state-of-the-art BASE-R and BASE-RC models. And you can see here in the graph that the BASE-R model by nature tries to assign for every locus that you consider whether it has a small or increasingly larger effect by assigning its effect to different distribution with increasing variance. So to put this in a schematic overview, so the BASE-RC model is an extension for that, which I explain on the next slide. But the question really was here, what if we have increasingly more complex annotations and multiple sources of them? How can we properly consider these in these models? So just to show this in a bit more of a schematic overview, the BASE-R model, what it does by default is that for every SNP, it assigns the effect to 4 possible distributions with assign increasingly more variance to the SNP itself. So that means that we only have those 4 distributions, and they apply to all the SNPs in the genome. The BASE-RC method extended that and said we're going to use the same modeling principle, but we're going to do it for a limited number of different annotations. So all SNPs would beforehand be assigned to a particular annotation class, and there'll be a limited number of them. And within each of those classes, we would have those 4 distributions where the effects would be sampled from. So just as an example here, let's assume that we have 2 sources of annotation, and one is GWS hits, and the other one is the QTL, the animal QTL database. And then this principle works pretty nicely in this regard, but what happens if we get increasingly more functional annotations, such as was the case here in GeneSwitch? So just adding here a few as an example, and what will happen at some stage is that SNPs will qualify to be assigned to multiple annotation classes. So then how are you going to go about to assign them to one of those? So that was the development that was done within the GeneSwitch project, was to extend this base RC model into 2 different variants. The one is the base RC pi method, where the model actually decides between if one SNP can be assigned different annotations, then the model decides where it actually should end up in the end. Whereas with the base RC plus model, it can assign a SNP to multiple annotation categories and sample its effect from multiple distributions in both cases as well. So this has been applied. First of all, this was on a big data set of a bit more than 1,000 individuals. 2 traits were evaluated in this case. In the plot you can see on the left there's an average daily gain, and on the right there's a back fat thickness. So what the bars here indicate is the difference in accuracy between those additional, the base RC, the base RC pi, and the base RC plus models over and above the standard base R model, which has only this one set of distributions. And you can see that all 3 models in this case for average daily gain actually found a marginal increase in accuracy, whereas for back fat thickness on the right, no such consistent improvement was observed. And then based on these results, you can dive further into looking into the different QTLs, assigning them to different traits, and actually see which particular trait in which particular trait groups were the QTL that had higher variance associated in these models. You can see that here in this plot. So in terms of prediction, we saw some modest gain for one of the traits evaluated. This was not consistent, though, across the traits. And for the interpretation, it really, this base RC pi model can help to identify the most informative annotations to boost QTL mapping. So just to summarize this bit, this has all been implemented, those 2 new models in the base RCO software. It was on the slide, but I didn't say this previously, which is freely available, so anyone that's interested, you can just go to this, to the GitHub site, get the package and try it on your own data. And what we saw is that with the base RC pi model, it can assign this informative annotations to multiple annotated SNPs, while accounting for the uncertainty in the prior knowledge. So beforehand, you don't really know to which of these annotations should it be assigned, and the model can help to make a decision on this. Whereas the base RC plus model, it can assign it to both, and the SNP can have effect in multiple categories, and this really helps also to upweight those SNPs that are assigned multiple annotations. So there's some fairly modest improvements in accuracy observed, and this was not necessarily consistent across all the traits. However, it was useful also, particularly what was seen in simulations, to help refine QTL signals. And it can also help to refine strategies for constructing annotation categories or deciding on which annotation categories are most useful to be used in prediction. So also what we looked into in addition to the Bayesian models is to compare machine learning models versus more standard linear models that are being used in genomic prediction. And in this particular situation, we used data that was available from a diversity outbred mouse data set. So the reason to use this data was that it was available early on in the project, and also because it has, for this data, there's a lot of recording on phenotypes and also individual gene expression, which will come in one of the later slides. So when we did genomic prediction in this data set, and we looked at very many different traits, I'll show you the results for 10 of them here. Because this population was so well characterized, we were able to classify for each of a trait whether it was really a polygenic trait or whether it was there was a lot of epistatic interactions underlying the trait. So if we look at those first 7 traits that were polygenic, what we saw is that the GBLUB, so the standard linear model used in genomic prediction, outperformed the machine learning model, the gradient boosting machine that we tried here. So that sort of makes sense, because if traits are very polygenic, the infinitesimal additive model probably applies, and that's exactly what GBLUB tries to model. Conversely, if we looked at a set of traits where epistatic interactions were known to be important, we saw that there was actually a benefit of using machine learning. So machine learning can go beyond this strictly additive model and actually model implicitly interactions between loci, and it seemed that it was able to do that and to give some additional prediction accuracy. So that was really nice to see that the results lined up closely with what we had expected. Then we continued working on this data, but in this case we had an additional source of information, and this was individual level gene expression measured on these mice. And as you can see here in the timeline with the red arrow at the very right, the gene expression was measured at 26 weeks of age, and it was measured in the liver. And here I'll show you some results for if we use this as gene expression as additional information to make prediction models to predict body weight at 10, 15, or 20 weeks of age. So this has all been published, as you can see here. So here in this slide, I first show the variance explained in those different body weight phenotypes in 10, 15, or 20 weeks of age. First I show what is the variance explained by only using SNPs, only using genotypes in the model. So you can see that in this case, so these are actually heritabilities, they range from 35 to 42%. Then if we used the gene expression data, their percentage of variance explained range from 68 to 75%. And it's important to note that, remember, the gene expression was measured at 26 weeks of age. So if you were predicting body weight closer to the moment of measuring gene expression, we were able to, with that, explain more variance in the body weight. And then finally, we built a model where we tried to assign as much as possible phenotypic variation to the SNP genotypes, and any unexplained variance could then be associated with the gene expression. And that's what you see here at the far right. So in this case, at all the ages, the amount of variance explained by the SNP is very stable, and it's very similar to only modeling SNPs. That's simply because we had parameterized our model to do exactly that. And then you can see that the gene expression on top of that explains increasingly more variance the closer we are to the moment of measuring the gene expression. So some take-home messages from this work on model development, but also evaluating them on these data sets as I presented. If we look at genomic prediction accuracy or across different trait architectures, then what we saw is that for polygenic traits, the standard GBLUB model typically is better because it matches the nature of the data better. However, if we have traits with known epistatic interaction that play a major role, then the machine learning models were able to outperform GBlob. Then about the individual gene expression that we used in the mice data set. So results showed, this resulted in more explained variants than only using the SNP genotypes, and in fact the individual gene expression explained a lot more variation than the SNP genotypes did. So there seems to be, it also helped to boost accuracy, so there seems to be really something in this. Looking towards an implementation in practice, there's quite a number of issues like current costs may be prohibitive, and it's also very questionable, you know, when do you have to measure it in time, which tissue, and you know, how do you standardize that all the animals sort of are in the same situation when you measure this. So there would be some hurdles to take there before being able to properly implement this in practice. And then the added value of using annotations in genomic prediction. So for that, we successfully developed this base RCO package, which is available on a GitHub. And this is also something where we will come back to in later presentations. The expectation of this is that using that will be particularly useful if we have high resolution of SNP genotypes, because we can get closer to the causal variants. And then when we are using this, we later sort of came to the idea that probably it's really important that we integrate all these functional annotations to make it easier on the model rather than having them all separately. So that's also what you will see and hear about later. That's all for this part.
Good morning, everyone. My name is Maria Ballester from IRTA, and I will explain the work that we perform in task 4.3 related with the identification of transcriptional regulatory variants in pig, duodenum, liver, and muscle tissues. Over the last decade, genome-wide association studies have identified thousands of genomic variants associated to complex traits in farm animals. However, most of these variants fall in non-coding regions of the genome that are enriched in regulatory elements and may impact gene expression. In this sense, gene expression is considered as an intermediate phenotype, and it is suspected to be closely related to the end-trained phenotype than the genotype, and closely linked to genetic variations than conventional phenotypes. The genetic loci that explains a fraction of the genetic variance of a gene expression phenotype are called EQTLs, and they are commonly identified through expression genome-wide association studies. Therefore, the objective of our study was to discover genetic variants that could be modifying gene expression levels across duodenum, liver, and muscle transcriptomes using expression genome-wide association studies. To do that, we collect samples from 100 individuals of Duroc, Landrace, and Last-White populations at the slaughterhouse. And here you can see the methodology that we use to analyze the whole genome seq and RNA seq from all of these samples. After filtering, we obtain 25 million variants from the whole genome seq, and almost 14,000 genes, 13,000 genes, and 12,000 genes expressed in the duodenum, liver, and muscle tissues. The eGWAS analysis was performed with the fast GWAS tool using the GCTA software. Now I will pass to explain the main results obtained in from our analysis. The A Venn diagram represents the number of expressed genes among the three tissues, almost 15 1,000 genes. And you can see in the middle the number of expressed genes that were in common in the 3 tissues. The B Venn diagram represents the number of expressed genes with at least one significantly associated variant. The AGWAS report 14 million significantly associated variants, and the muscle was the tissue with the greatest number of unique significant associations. This result may be due to the increased selective pressure that this sample, this tissue, has experienced during over the last century. Liver and duodenum share the greatest number of significant associations, and this result may be due to the shared embryonic origin of both tissues. From the significantly associated variants, we only identify 3% of polymorphisms associated with the same gene across the 3 tissues, indicating a high proportion of identified genetic variants with tissue-specific regulatory potential. Among the significantly associated variants, we identify, on average, 56% were cis regulatory variants, since they were located within 1 megabase from their associated gene. For the EQTL region annotation, we only keep those EQTL regions with at least 2 associated variants, including 1 megabase up and down of the significantly associated polymorphism. Also, we annotate the EQTL regions as cis or trans, depending on the position of the EQTL with their associated gene. As explained above, if the EQTL region was within 1 megabase of its associated gene, was annotated as cis EQTL. Conversely, if the EQTL was located farther than 1 megabase or in another chromosome from their associated gene, was annotated as trans EQTLs. We identify 26,000 EQTL regions, and from these, the 22% were cis EQTL regions. Cis EQTL regions comprise more significant polymorphisms, but trans EQTL regions were more abundant. When we assess the distribution of cis regulatory variants, we obtain in the cis EQTL regions, within the cis EQTL regions, we obtain a similar pattern of distribution in the 3 tissues. Here you can see, as an example, the duodenum tissue. Gene locations with the greatest number of cis regulatory variants were located within 100 kilobase upstream the transcription start site and downstream the 3' UTR region. We identified 23 top regulatory variants found in common between the 3 tissues, and of these, 6 were identified as top cis regulatory variants. In this table, you can see the position of these cis regulatory variants and the gene associated with. Also, we performed the analysis of the identification of hotspots and top hotspots regulatory polymorphisms. The hotspot was defined as a significant polymorphisms associated with the expression of 10 or more genes, and top hotspots were defined as the most significant polymorphism associated in at least 10 EQTL regions. In this image, you can see the definition of the hotspot and top hotspot. Here in the table, as a summary, you can see the number of hotspots and top hotspots identified in each tissue. And we identify 100 hotspots in common across the 3 tissues. Also, from these top hotspots, regulatory polymorphisms, we identified 23 top cis regulatory hotspots associated with 11 genes. Liver was the tissue with the greatest amount of putative regulators. As an example, we identified the CHD7 and CHD8 genes. These genes are ATP-dependent chromatin remodeling enzymes that are involved in gene transcription regulation. Also, in order to evaluate the modulation of the trans-associated genes through changes in the expression of the regulators, we perform a gene co-expression network analysis, and we see that almost all the regulators were co-expressed with their trans-associated genes. Finally, we also perform a motif discovery in cis regulatory SNPs, and to do that, we obtain the sequence, the 20 base pair sequence of the position of the cis SNP, and we use the MEM suite software to do this analysis. We discovered two motifs that were recurrently found in the 3 tissues. The first motif was a poly-A short tandem repeat. This adenine short tandem repeats has been identified, is the most common sequence found in humans, and may have an impact in the transcriptional, in the transcription of the genes through affecting the poly-A site, and also have an effect over the nucleosome and transcription factor accessibility. The other motif that we discovered was usually found in the 5' flanking regions of the associated genes. It seems to disrupt a ZPG methylation site because in fact the mutation is a G, 1-idin cytosine. And this motif was found or has been found in human promoters. As a conclusion of our work has allowed the identification of genetic variants and candidate genes that will help to understand the molecular mechanism behind gene regulation and their potential effect over gene expression and end trait phenotypes. And this functional information could be useful to improve the accuracy of genomic prediction in pigs. And I think that Bruno will speak about this later. These results has been published this year in the GWS Science Journal. And I would like to acknowledge all the people that collaborated with in the consecution of this, in this work, but especially to Daniel Crespo Piazuelo, that was a postdoc that worked in this data and, in fact, performed all the analysis of the EQTLs. Thank you for your attention.
Questions for Maria. So the first question is, you saw that for your top SNPs, quite a lot of them were indels. Is that just a coincidence? Or your top SNPs, many of them were indels, so not SNP polymorphisms. Was that expected, or? Okay. and okay.
Otherwise, if you can come over here, then the online people can hear you as well. Oh.
Oh, okay.
Okay.
I don't know if.
And maybe a second question. So you have these hotspots. Did you also see that if you have a gene that was also like in a cis EQTL that were potentially regulating the trans genes, did you also find if they had any transcription factor or cofactor annotations? Because that would be kind of the hypothesis, right?
In fact, the hotspots are transcription factors that I indicate are all transcription factors or cofactors.
Are they all transcription
Yes,
factors or
yes.
cofactors? Okay, no, then that's clear.
Any other questions? Actually, I had a question, Maria, because you mentioned the you report on two motifs. So how could that be utilized in genomic prediction, or what would be the next step to follow up from that?
The two motifs are two SNPs that are in the motifs, and I think that you can use the polymorphism or to put in the models to check if you have more.
So then you would still have
it's
to...
like, I think that you can use all the EQTL results, all the polymorphisms that we, and give more, I don't know, more weight to check if you increase the prediction or not. I think that in this way.
Thank you. No online. Oh, there's one. Hervé.
So for Maria, when you have the top hotspot, you think that it's just targeting one main gene, and after this main gene will affect other genes, or you're targeting an answer that will directly affect the expression of many genes?
In this case, we have two top hotspots that are in cis or top hotspots that are in trans. The trans hotspots maybe are enhancers, and the cis top hotspots are in the promoter regions or within the gene. And I think that is a change in the transcription factor that is impacting the trans-associated genes. I don't know if yes?
Okay. No online. No. Okay.
Okay. So good morning, everyone. So I'm Bruno Perez. I'm a geneticist for Hendrix Genetics, and I'm going to present our latest results from including function annotation into genomic prediction models. Okay, so just some background first. Well, in this investigation, our objective was to include function annotation in the form of annotation maps into genomic prediction models. And then how did we arrive here? So first, Mario and Marco talked about genomic prediction and well, our past experiences with including only one source of information, one layer of function annotation into genomic prediction models. And there we saw very highly trait-dependent results. And also Mario also mentioned that, well, then we thought, okay, maybe the best way would be to integrate these many layers in one whole approach. Then how can we do that? I'm pretty sure we can do that in many different ways, but the FAANG scores that I show in the slide, they show a very nice, well, it's a nice framework to include all these function annotations together. So the FAANG scores were published in 2019, first by Xiang, the paper I show below. And it's a framework to score SNP variants according to how relevant they are to across function annotation maps and across a set of phenotypes that you have. I'm going to show just quickly how the FAANG score calculation works. So you have basically 3 steps. First step, in the first step you define your function annotation map. So they're basically positions or regions in your genome that rise from results of these annotation layers. And of course they can be different across different function annotation maps. It can be either based on p-values or other measures. Then with these maps in hand, what we do is we can build, we go back to whole genome sequence level. We extract these SNPs that pertain to the function annotation map, and we use them to build a genomic relationship matrix. And we call it a target genomic relationship matrix. And we do it across every map. And later on, we fit together with what we can call a HD genomic relationship matrix. So we fit two genomic relationship matrix. One is the target functional annotation map across every map, and another one more to absorb the background noise, let's say, and to be able to capture the, well, more accurate proportion of variance explained by the target GRM. With that, when we accumulate these scores across traits and functional annotations, then we can rank our variants according to how influential they are. So then we with this in mind, we built a pipeline just to be more, well, the whole focus of the pipeline would be to be more dynamic in terms of what functional annotation maps we could include. So then basically we start with whole genome sequence, functional annotation maps, and our phenotypes. Then we use Bink software to subset across the functional annotation maps, subset from whole genome sequence the SNPs that we want. And we use, we build the GRMs, target GRMs, using Calc GRM software. We also build the HD GRM, use it across all functional annotation maps. Then we decorrelate our phenotypes, and we estimate the proportion of variance explained by each GRM across all our traits. So the data that we used for this investigation, so we had 100 landrace individuals with whole genome sequence available, and we used these 100 animals to impute the whole landrace, well, the genotyped population. So we had around 25,000 individuals with imputed whole genome sequence, and these summed up to around 15 million SNPs. We used 6 different traits. You have just a description of these traits, how many observations we had for the reference, for training, how many observations we had for validation, and the heritability of each trait. They're coded here, but we had around, well, between 6 and 20,000 records per trait. Together with that, we put on now a vast collection of functional annotation maps. If we compare to the paper from Xiang, they used around 15 functional annotation layers. And now, in our study, we summed up to 32 functional annotation maps. And they come either from consortium data or other publicly available data. So I'm going to go quickly through, well, a little bit more in detail for the consortium data. Of course, we use data from GeneSwitch, and it's nice to know that all the maps that we use there, and I indicate in the slide, were previously presented here, one way or another. So we have the EQTLs, so what the work that Maria presented from IRTA. We have differentially methylated regions, the work that Martijn presented from Janni, Wageningen University. Then the ATAC-seq presented by INRAE, and the CheapSeq that also worked with by Martijn. We also use data from the PIG-GTX, FARM-GTX consortium, which is a big consortium, has a lot of data. I would say the main difference between the two is that for GeneSwitch, we have the developmental stages. This is an aspect that the PIG-GTX does not have, so GeneSwitch brings in addition to it. I'm not going to go into detail, but these are the maps that we use from other sources, mainly publicly available through publications, papers. Okay, so after we run the pipeline, we have Fed scores, and we can estimate what we call SNP-based heritability. So this is basically for every target GRM, for every GRM built by the functional annotation maps. If we divide the heritability estimated by this component by the number of SNPs in the functional annotation map, then we arrive at a SNP-based heritability. It's an average percentage of errors explained across all our traits. So on the right, you see for our results with the 32 different maps and pointing the GeneSwitch maps there. And some bullet points for conclusions there. So first thing we see, molecular QTLs score very highly. So the green ones in the graph. Among them, the gene switch, the EQTLs that we just saw the presentation from Maria, and also from PIC-GTX. Also, you can see there are small and big standard deviations. Standard deviations here would indicate trait-specific, I'd say, effects of the functional annotation. So, for example, the first one, the highly scored, it has a very high standard deviation, and this is mainly due to the map performing quite well for one trait, but not for others. And it seems to be more common among the molecular QTLs. Also, one interesting thing is that, as you can see, the colors in the graph show different groups, types of functional annotation maps. And nice to see that the similar, the colors group together. And just one final topic on the conserved sites from the previous paper on Xiang. It scored very, very highly for cattle data, for dairy cattle, but it does not seem to be the case in our example, but it also may have to do with the fact that the traits in dairy cattle, included in dairy cattle, might have been more subject to natural selection for long periods. Traits like stature, morphology traits, and which we don't have for pigs, not in this example. Then if we zoom in, so the graph you see here is just the same graph I just showed, but now every trait has its own dot. And you see that across the functional annotations on the x-axis, different traits are scored differently, right? And we see, especially for trait 3, it's often the highest scored across our functional annotation. So there is some predilection to this trait. So this trait seems to be more affected by these functional variants. And then we see that both functional annotation map and the phenotype set that we use are probably influencing our results. If we're using 6 traits, but if we use completely different traits, probably our FAT scores will also change, of course. Then we went to validation, so we calculated our FAT scores. Next question would be how to validate them. Do they bring something to genomic prediction? So what we did, we planned a comparison between the highest and lowest FAT score variance, and also the highest variance against a randomly sampled but same density SNPs. Basically, 2 questions: can the FAT scores indicate informative SNPs? And can the highest FAT scores outperform just a random placement of SNPs for genomic prediction? First result to see is on heritability, so percentage of variance captured. And we are working at 1 million SNP level, so we have 3 different groups. The low 1 million, so the lowest FAT scores, 1 million lowest. Randomly sampled 1 million SNPs, and highly, so the highest FAT scores, also 1 million. So first thing we see, the low FAT scores, they always capture less genetic variance than the high, and also then randomly sampling SNPs. And you see that the last column on the table indicates how much, what's the difference between the low and the high FAT scores. And also notable that the high FAT scores are not necessarily capturing more genetic variance than a random placement of SNPs. Then we go to predictive accuracies. Can we translate these FAT scores, high FAT scores, to higher predictive accuracy? What you see on the right is the same traits, the same 6 traits, and the same 3 different panels. So low, 1 million, random, 1 million, and the highest FAT scores, 1 million. First thing we see is that the low FAT scores yield the lowest predictive accuracies across all traits. So it's approximately 15%. Lower than the randomly placements. That the high scores yield either very similar or slightly higher predictive accuracy when compared to random placement of SNPs. But the good thing is that, different from what we saw before, when we fit only one layer of functional annotation, is that the high FAD score never seemed to deteriorate predictive accuracies. And when we look at the traits individually, we see either no increase or we see from 1 to 4% absolute points increase depending on the trait. So our next step was, okay, we can subset our SNPs from whole genome sequence, and we can use FAD score for that. But what if we can inform the model a little bit more? So if we can go further, and well, we have FAD scores, even if after we subset our top 1 million, we can still discern between them because they have their own values. So what if we can inform the model, okay, if the highest of FAD score variance is informing more than the lowest of the highest, right? So then we derived weights from the FAD scores, and we used them to weight the SNPs when building the genomic relationship matrices, and you just see the distribution of these weights as FAD scores and as weights on the right. However, when we use them and fit them into a predictive model, a genomic prediction model, so now what you see in the graph is the gray and the blue bars are exactly what I just presented, but now I included the yellow and the orange bars, and they indicate including FAD scores as weights in the GRM, building the GRMs, either on a linear or a quadratic form. And you see that across the traits, it does not seem to bring much to predictive accuracy. So take-home messages. First, well, we developed a pipeline. It's a very general pipeline. It fits different species. It's sort of a plug-and-play pipeline. To calculate FAD scores, we can use publicly available data, proprietary data. It all depends on putting it all in the format that can be inputted to the pipeline. From the results, we see that molecular QTLs, eQTLs, mainly can seem to be the most informative functional annotation maps, but also the more trait-specific ones. They seem to work very well for some traits, but not necessarily for other types of traits. From our results, we see that the FAD scores can help to improve predictive accuracies and pig breeding, but using them as using FAD scores as weights are not necessarily resulting in an increase in predictive accuracies. And just thinking about possible limitations there, maybe by imputing big blocks due to LD, basically, we lose some resolution in the discovery part, which will have consequences to the prediction part. So we are able to point out to relevant regions, but not necessarily differentiate between two SNPs, what is the most relevant. Just quick on next steps. One thing we discussed was most of our animals were genotyped with one map, the commercial map that we use in routine. So this means that for these animals, every genotype for the variants that are included in the 50K commercial map, they are not imputed, they are the actual genotypes. And this brings some inflation because it have no noise around it by the imputation, since they are not imputed. So we were just a bit concerned on can this bring some inflation in the results. So this is what I'm testing right now. And of course we are pursuing a publication just to report on the results I just presented. Yeah, and I'd like to thank everyone involved. The data that was provided to us from the teams that worked with the raw data. Yeah, thank you.
Okay, so today I will present to you fine mapping and functional annotation of growth-related QTL and GWAS loci in chickens. And the lines in question are the Virginia chicken lines. They've been divergently selected for one single trait and one single trait only, body weight at 8 weeks of age. And this is the trajectory per generation for the high line. And this is the trajectory for body weight for the low line over 57 generations. At generation 40, these two lines were intercrossed to generate an advanced intercross line that was kept for 17 generations. And as you can see, this is the phenotypic mean, so the advanced intercross line. They converge to approximately the mean of the parental populations, indicating a highly polygenic architecture for the trait in question, which was body weight at 8 weeks of age. And these lines were generated by Paul Siegel at the Virginia Polytechnic Institute. So for the statistical genetics part of this project, we sequenced around 3,300 individuals from the advanced intercross line using Illumina at low coverage. And we sequenced the parental high and low individuals at high coverage. Then we called variants and used imputation to predict haplotypes inherited from the high and low for the advanced intercross line individuals, as well as collected the body weights also at 8 weeks of age. And then we proceeded to do association analysis. And we did this both by eQTL screens and using GWAS method. So this is how QTL and GWAS peaks look like over the entire genome. So in red, you have QTL peaks, and in dotted gray, you have GWAS peaks. And as you can see, they do not always agree perfectly. And this is expected because QTL and GWAS have some differences, because QTL assumes a near fixation for opposite alleles in the founder lines, where for GWAS, there is no such assumption. So if we compare the minor allele frequency in the advanced intercross line, we can see that the minor allele frequency for GWAS, significant GWAS markers, they go down to quite low minor allele frequencies, whereas for QTL, they reside much higher. And for the ones shared between markers and QTLs, we have very high minor allele frequencies on average. And if we plot this instead against FST, genetic divergence between the founder lines, we can also see that the ones, so markers, they are not necessarily, so the GWAS, significant GWAS regions are not as often existing as divergent SNPs in the parentals, whereas the shared ones and the QTL ones more often reside here in the very high divergence range for the founder lines. If we characterize the genetic divergence between the founder lines, and we do this by two different methods, one quite stringent, where we require delta allele frequency to be 1, completely fixed for different alleles. We can see that these are distributed in about 2,000 10 kb windows. So the genome, summing up to 69 megabases. And if we relax this criterion a little bit, we reach a figure of about 18% of the genome are nearly fixed for opposite alleles, or have at least one marker which is fixed for opposite alleles in these lines. So for the functional annotation part of this project, we used various gene switch sources. We used gene and non-coding RNA content in GWAS and QTL regions, whole genome bisulfite sequencing data to calculate methylation in CpG clusters, coding variants, high-C data, gene expression level from the isoseq data, as well as attack sequencing data. And in addition to the gene switch data, we also used data generated from different sources. We used the variant effect predictor, we used SIFT, and we used evolutionary constraints based on philo P scores based on the 77-way vertebrate genome alignments. And when intersecting genes and non-coding RNAs with this data set, we noticed that the QTL and the GWAS regions contained about 2,000. So the GWAS regions, when we class a significant region as having SNPs that have a minus log P of above 4, we get 2,000 genes. When we instead use the minus log P above 6, we get 734 genes that are protein coding. And for QTL regions, we get about 3,000 protein coding genes. And as you can see in this top figure here, we have consistently higher amounts of genes per megabase within significant GWAS regions in compared to regions outside of significant GWAS regions, both for the P4 and the P6 level. And you can see the corresponding increase also for non-coding RNAs. And albeit not as strong enrichment, there is still a small signal for enrichment of genes within QTL regions. And if we show this as an enrichment ratio instead, we can see that we have about 1.4 times as many genes per megabase in the significant GWAS regions, and about 1.6 times as many genes in the significant GWAS regions when we increase the significance threshold a little bit. As I mentioned, we also use the variant effect predictor and SIFT to classify SNPs into various classes. And the classes we used were synonymous, missense, and high consequence based on the variant effect predictor classification. And SNPs annotated as missense were further annotated by SIFT as deleterious or deleterious at low confidence. And variants in the high category were all affected to predict or predicted to affect splicing or stop codons. So we counted how many of these exist in significant GWAS and QTL regions and checked if we had a higher proportion than we would expect based on synonymous variants, since synonymous variants are expected to be neutral. And in this table, we have the sums of SNPs identified in these different categories. And in parentheses, there is the relative enrichment compared to the expectation calculated from the synonymous counts. So we actually got a lower value than expected from the deleterious missense mutations within GWAS regions, as well as QTL regions. But when we look at the high impact variants, we see that we actually seem to have an enrichment of them, albeit at very low counts. If we use delta allele frequency as an additional criteria between the founder lines, we can see that this doesn't help us much. But we think this is a possibly interesting signature where we have an enrichment of splice variants or variants predicted to affect splicing and missense variants within GWAS regions. On chromosome 4, we identified 4 out of 6 regions identified to be epistatic QTL. And they cluster in a relatively small region on chromosome 4 of approximately 26 megabases. And when we were looking from the high C data generated within GeneSwitch, or actually capture C data, here shown is hind limb muscle at stage E15. We can see that where the QTL signal peaks up here, we do seem to have something resembling a TAD. And if we zoom in a little bit, we see that there is plenty of off-diagonal contacts connecting these regions, which would theoretically make it feasible for cis effects being involved in conferring this epistatic nature of this cluster of epistatic QTL on chromosome 4. As I mentioned, we also investigated methylation in CPG clusters and found that CPG clusters in the muscle of newly hatched chicken have a lower methylation within GWAS regions. So we identified CPG clusters using the CPG cluster algorithm, and then we calculated the average CPG methylation across tissues and development stages. And we checked how these distribute with regard to average methylation in associated and non-associated regions. And by associated, I mean associated with the trait body weight at 8 weeks of age. And the most significant difference was a 17% reduction in CPG cluster methylation in muscle from newly hatched chicken. And this was for the higher stringency definition of GWAS peaks. But if we use the lower stringency definition of GWAS peaks, we reach a 13% reduction in methylation. And you can see this here, where regions outside of the GWAS P4, they have an average methylation of around 0.55. And CPG clusters within, they tend to have an average methylation of around 0.4. And here is the data support from a comparison of several tissues and stages. And we can see that the one tissue and stage which stands out is hind limb muscle of newborn, newly hatched chicken, where we also observe the lowest or the highest reduction, the largest reduction in methylation. And this is interesting because it could indicate that genes activated in muscle to promote muscle growth after hatch are enriched in these regions. And this is actually supported if we check which functional terms are associated with genes within GWAS regions. And here we also identify categories such as myofibril assembly and muscle cell development as significant. So we do seem to have an enrichment of genes affecting, involved in muscle development pathways within these regions. We also investigated whether we had an enrichment of attack-seek peaks in GWAS regions. And we did this in a way where we checked for enrichment of attack-seek peaks alone or attack-seek peaks coinciding with positions identified as being under evolutionary constraint using phylo p-values or phylo p-values alone. And the plot below here shows the enrichment within GWAS regions. So for attack, we don't have an enrichment either at P6 level or at P4 level. But if we combine attack with phylo p scores above 2, 3, 4, and 5, we do see that as we go up to relatively strong evolutionary constraint, we do see a small enrichment. But as we go down to extreme constraint, we lose this enrichment. And as you can see here, we're talking about very few SNPs in these categories. And when we compare this to what we get if we use phylo p alone, we see that phylo p alone seems to give a stronger enrichment in these regions. So the enrichment we see by combining attack and phylo p does not surpass the putative enrichment we get with phylo p alone. So in conclusion, we identified various genetic associations to body weight in the AIL line. And we identified around 20,500 SNPs that were significantly associated with body weight, distributed across 42 distinct regions. And we relaxed this criterion in some of these analyses to see if we caught additional effects. And in QTL analysis, we revealed 42 QTL regions and 6 epistatic hub loci clustered on chicken's chromosome 4. And that QTL and GWAS peaks do not overlap to a high degree due to some differences in the nature of these two methods, which makes them complement each other. For the functional annotation part, genes and non-coding RNAs were enriched in QTL as well as GWAS regions, particularly for GWAS regions. And that in muscle of newly hatched chicken, we saw consistently lower CPG cluster methylation within GWAS regions, but also saw this trend for QTL regions. In variant effect predictor analysis, we identified an enrichment of SNPs of the variant effect predictor high severity category, which includes variants with potential deleterious effects on genes, and these were enriched in GWAS regions. And finally, that significantly associated SNPs were not enriched for RNA-seq peaks, but they did show a slightly stronger sign of enrichment at evolutionary conserved sites. Thank you.
Very good. Thank you very much, Carl-Johan. I think we keep the questions until the after the final speaker of the session. So we move on to the next speaker, Andreas Kranis, who will talk about subsetting GWS SNPs based on function annotations in chicken.
Perfect. Okay, so hello again. I'm going to talk to you about the work that we've done in package 4.6 with Irini and myself and other colleagues. But before starting, I just want to give some perspective on perfect. Okay, sorry, needs to push hard. About how things evolved in the genomic prediction and the application in breeding programs. So chicken was the first farm animal that has been sequenced. The sequence has been available since 2004, and then there were various panels released that coincided with the developments in the technology, and the density kept increasing. The main challenge, of course, was the cost, especially in poultry breeding programs, where the value of the selection candidates is small, and we can't justify the cost. So back in the early days, it was costing, including processing, almost $200 or $300 to genotyped one animal, and that was not sustainable. However, technology improved, and we were able to increase accuracy. And that was important because at these early stages, density was deemed as one of the most important factors when it comes to get more accuracy. And obviously, the density was important because it allowed us to capture LD blocks. However, quickly, we got into a stage having 600,000 SNPs in the panel. And that meant that we could start looking at the impact of the density and regarding the accuracy. So back in 2014, we looked at that, and what we've seen was that going beyond the few thousands or few tens of thousands of SNPs didn't offer a lot in terms of accuracy, and what was more important was the training size, the size of the training data, which again, I mean, gave some indication that the structure and the relationships are also very important. And again, this is not, these are chickens, these are closed populations, so they are not exactly the same as, for instance, what our colleagues in human genetics are doing. So again, we know, of course, that there is an ascertainment bias in terms of which SNPs go into the panel. Obviously, we favor SNPs that have higher minor allele frequency. I have been involved in building the 600K chip in chickens, and that was, again, a big challenge because you have to bring together the layers and the broilers, which are completely different. So all sorts of limitations of the SNP panels. And that's why moving to whole genome sequence data gave a lot of promise. The ascertainment bias is not anymore applicable. Hopefully all the variants are in the data. Well, when I say variants, obviously SNPs. But of course, in that case, our job when it comes to do genomic prediction becomes more difficult because now we have millions of potential predictors, and therefore, again, we are back in square root one where there's too much noise in the data. So obviously, our aim is to improve the noise to signal ratio, and that's exactly the promise of using whole genome sequence coupled with improved annotation. So that allow us to not only incorporate this annotation, but also try different combinations and see the impact of having this information at hand. In order to do that, it sounds simple, but we can decompose it to three main steps. And first of all, we need to build this large repository of the whole genome sequence data, annotate the SNPs, and run the genomic evaluation. So when it comes to building the whole genome repository, we had a quite big data set. Again, what we are working is a closed population, and it's a breeding program that has been evolving, but also closed for many years. So obviously, if you think about how the gene flow happens in the data, we were actually able to sequence most of the parents that through them the genetic progress go, and because of course everything is linked, even the ones that we haven't sequenced, probably they have some common ancestor here. So by projecting these haplotypes, we pretty much have captured most, if not all, the genetic variation at a given point in time. So we had 518 30x individuals sequenced. And then we had a pretty large data set consisting for almost 140,000 individuals for which we had 50K genotypes available. So the simple thing was to impute them, right? I mean, that's not, it's one of those things that is easier to say and more difficult to do because obviously of the sheer size of the data. We had 12.8 million SNPs, and our imputation pipelines worked quite well. We got an accuracy that was exceeding 0.95. Again, with a caveat that obviously rare alleles have lower accuracy, and this, I think Bruno already mentioned that this could have an impact on the results, but that's what we get from imputation accuracy. And obviously, in order to do that, and build, effectively we had to build a database that was holding 140,000 whole genome sequenced animals. You cannot do that with off-the-shelf tools, so we had to build specialized libraries and specialized data handling tools in order to be able to subset any set of animals and SNPs in order to pipe those analyses. So again, this is background work that is necessary. It takes time. It's not something that brings glory, but is essential, especially when we talk about this kind of data. And when it comes to adding the annotation information, again, there were multiple sources where we could take. Obviously, being part of the GeneSwitch project, we had access to the outputs of work package 2, and in particular, the improved annotation that Sebastian talked yesterday. Obviously, all this work using ISO-seq and discovering new genes, this was incorporated. So we annotated all our SNPs using this, exploiting this. There are also other outputs like the attack-seq and the methylation patterns that potentially could go into that. But again, there are also resources available, like the animal QTL-DB, which is part of the chicken QTL-DB, which again, they collect all the published QTLs. So again, we could annotate all our SNPs and mark the SNPs that are within those QTLs. And finally, there are also third-party sources, particularly this one here, the conserved non-coding elements, which again is an effort that looks into genomes of different species and try to find the regions that are conserved among these species, the non-coding regions, which again, the fact that the region is conserved could signify that there is a functional importance to that. And then again, we can see the SNPs that are within those regions and use those bioinformatics scores that are trait agnostic, but still useful to us. Again, because of the different sources, we had to build a flexible database to incorporate that information. And this database is not about just using the information that is available, but others is also flexible to allow us to bring more information if required. So again, this is another important parameter to think when we do projects like this. And finally, the third component is bring the phenotypes and the models. In this case, we focus on body weight, which is, if you like, the equivalent of human height, where a lot of studies are already available as well, both across farm animals, but especially for chicken. Body weight is a good trait because we maximize the number of phenotypes. So, in other words, all the sequenced animals have a phenotype. On the other hand, body weight is a highly polygenic trait. These lines have been selected for body weight for many, many years, either directly or indirectly through the other traits in the breeding objective. So again, the choice of the trait is debatable, and obviously it will have an effect on the results. And again, due to the very large size of the data, we had to apply different divide and conquer approaches, try to parallelize things as much as possible. So again, the idea was to use the different annotation information to group the variants into different categories and try to see how we can build GRM matrices that account for that. And yes, again, for speed ups, we could use ridge regression, which is equivalent to a GBLAB, at least when it comes to a univariate analysis. The variable selection methods are also important, like the base R and the base RCO, but again, due to the size of the data, these are iterative approaches. They use GIP sampling, which takes a lot more time to converge. And when it comes to the accuracy, this has been assessed as the correlation between the GBVs and the adjusted phenotype, adjusted for fixed effects. This is a trait that is not, it has its limitations. Again, we are not in the fortunate situation like our colleagues in dairy cattle that they use adjusted progeny means that are much well correlated to true breeding values, if you like. So again, these are limitations that has to do with the way that the breeding program is structured. But nevertheless, it's what people use. So again, it's not a deviation from what is done. So when it comes to annotation categories, as I said, we had different annotation categories. I mean, this is a picture from an ensemble where they saw each different category where it belongs. And we devised different in silico panels, right? So the first one was a cheeky one, right? It consisted of 45,000 SNPs, explicitly trying to get SNPs that are intergenic, and they have absolutely no annotation tag against them. They have no QTLs. They don't belong to the 50K, and also they don't belong to any conserved area, right? So this 45K, which was, by the way, I mean, we had, I think, 1.7 million intergenic SNPs that fall into that category, and those 45, they were equally distributed across the genome using LD pruning, so trying to capture just cover the genome, right? So this 45K panel, hopefully, is as less informative as possible when it comes to functional. And then we had, again, using pruning, splicing variants, exon variants, and then using, for instance, the terminology of ensemble, the high impact, which include splice acceptor and donor regions, and stop gain and stop loss, or moderate impact, which is our missense variants. As I said, we brought also information from the QTL database. And also, we did a GWAS in our data. To see whether we get peaks, right? And this was also prior information that was meant to be used. And finally, we had our routine 50K, right? Which is what we are using in practice. Before going to the results, I just want to show you how those panels look across the genome. And as you can already see from the colors, they are quite different. So first of all, the GWAS hits, right? They are 6,000 SNPs there, but of course, as you can see, the gray zones means no SNPs. Pretty much a very small portion of the genome is covered from those GWAS hits, which correspond to the GWAS peaks using an FDR of 0.1. Again, and these are 6,000, almost 7,000 SNPs, but they are really concentrated in small bits of genome. With similar number, the high impact is covering quite a lot of the genome. So although we have the same number of SNPs, in terms of how they are arranged in the genome is quite different. And then we have the intergenic, which, yes, by design, covers a lot of the genome, but there are some gaps, because obviously these are the functional bits, but then again, it covers the whole genome. So it allows us to capture Mendelian sampling. And finally, we have the 50K, which the 50K was not, obviously, it also covered all the genome as uniformly as possible, but it was favoring also coding regions and functional SNPs with the information that was available back then. This has been developed almost 10 years ago, and obviously the amount of functional information back then with now is not the same. Nevertheless, there is quite a lot of functional information in the 50K. Right, so let's see some of the results, right, and what we try to do. In this case, again, I remind you, had animals across time. We took random splits across all the animals. So it was an 80-20 approach. In total, 60,000 animals. And again, the purpose of that was to try to take random partitions to see also a bit how things differ. But that's not the typical scenario in animal breeding, because obviously the testing set is not, there are relatives around the testing set. And also, just a note, these are the same 60,000 animals that we use for GWAS. Starting with intergenic 45, the agnostic, if you like, panel, all the numbers here are relative to the 50K, right? The 50K, which is the routine. And as you can see, everything above 50K is red, which means that we have less accuracy in terms of, we are losing a bit of accuracy. And the worst category is the QTLs, the published QTLs, right? Which again, re-emphasize our struggle with QTLs, right? That we read a paper, they report QTLs, we cannot validate them. I've been through this excruciating exercise many times, and every time I lose more faith in that. Again, these are quite significant drops. Interestingly enough, the 45K does worse, which is good, but still captures a lot of variation. But the good thing is that starting adding information to the intergenic gives us something. So we go from, there's a trend across time that adding exons or splice or high impact SNPs gives us something. Now with the high impact SNPs, again, because they cover a very small bit of the genome, they give us something, but not a lot. Now, with the 50K, obviously, we are doing pretty well, but when we try to add the exon, we get some marginal improvements, which are not bad, right? I mean, they just give us a bit more. And what gives us even more is GWAS results. Now, again, this comes with a caveat that it's the same population that we did the discovery. So to summarize, I think that in this table, we see a few things that are important here. The fact that by adding, if you start from a panel that hopefully doesn't have a lot of functional information, by adding, you get more. And also, there is some marginal improvements when it comes to accuracy. Again, these are coming from GRMs with when we applied base R, for instance, the results were slightly lower in terms of accuracy. But again, the indication that we saw was that, for instance, when we were putting 50,000 SNPs, there were only 500 that were actually in the non-zero distribution, you know, the 4 prior distributions that base R has. So most of the SNPs were going to the first one, which is, again, signifying that there's not a lot of big QTLs, which is obviously something that we know. As I said, because we, in the first trial, it was the same population that we did the GWAS and the prediction. We just wanted to validate the GWAS hits on the 70,000 animals that were not part of the validation set. And then again, there are relationships, obviously, between those animals, but at least it is not the same. In that case, it was a forward prediction. So the testing, the validation set, were the progeny of the training set. In that case, the GWS alone is not, so if you build a GRM matrix only with the GWS hits, that's not great. But when you add those GWS hits to the 50K, we get a marginal improvement. And obviously adding the published QTLs just in case doesn't really give us anything. So in a way, we kind of shown that some of those GWS hits might be relevant. And actually, when we look at the heritability explained by those two GRMs, that has a heritability of 5.5%, and this one has a heritability of 19%. What is striking, and I don't have in this slide for the interest of time, is how different those GRMs look like. They are strikingly different, right? And this is a direction that we would like to look a bit more, what actually those GRMs mean and what do they capture. Finally, we just wanted to see whether we can get something more out of the power of the data. So in that case, we had a training set that was consisting of 105,000 animals, and we were trying to produce 10 consecutive cohorts of selection candidates. In that case, again, using only the exon gives us less accuracy, but when we combine the exons and the 50K, again, we get slight improvements in accuracy. I mean, these are marginal improvements anyway, but still, there is something there. So, as I said, those improvements are modest, but they are not. This is also something that has been shown from other studies with whole genome sequencing data. Again, thinking of what we were doing, we were using body weight, which is a polygenic trait. There is merit to think of other traits. The challenge, of course, is that although the breeding objective in broilers consists of more than 30 traits, most of those traits are quantitative and polygenic in nature, and therefore is not necessarily easy to find alternative traits. Having said that, I think that there is scope to look more into other characteristics as well. And I just want to re-emphasize the computational challenges, right? I mean, these are not trivial data sets, these are big data sets. We had issues with convergence in some cases, especially when we try to put more than one GRM in the same analysis, and also issues with time, especially when it comes to the iterative algorithms. And finally, I want to highlight things that not necessarily are highlighted usually, but building the capacity, being able to handle these data sets and develop that in large scale is not a trivial task, right? These are necessary steps to think how we can, what else we can do. And when it comes to that, yet again, I think that getting a variant-specific prioritization is important, relating the phenotype specific for the traits. We saw that perhaps for the first time, GWS has some value for us, but this is just the beginning when it comes to the variant prioritization. Trying the integrated scores like FAITH, we are working on that. Again, as I mentioned, there are some issues when it comes to convergence because of the size of the data. Which brings to the last point that, yes, a GRM is something that we want to look how the GRMs relate and how they compare with each other, but the variable selection methods will play a role, but need also to be improved in terms of speed and the ability to handle big data sets. Thank you very much.
I think with that, thanks again, Andreas, for a very elaborate presentation. We still have about 10 to 15 minutes left for general discussion and questions. So the question for Maria, Ola, nice talk. I have two questions. One, are some EQTL SNPs associated with traits of interest? That's the first one. For Maria. Oh, yes. So are some of the EQTL SNPs are they associated with traits of interest?
But we don't look to this at the moment.
Okay.
For traits of interest, no. For the moment, we only perform the EQTL analysis.
Yeah.
We don't look to the association with the traits of interest.
Yeah. No, we maybe to elaborate on that, we did try to look in the same three populations on traits of interest, phenotypic traits, but we decided not to continue on that because data was very incomplete and heterogeneous. So, okay. Second question for Maria is, what next? Will you validate some of these EQTL SNPs?
That is an interesting question because I like a lot to do experiments in the lab. And yes, we can validate some of these results by luciferase assays or by some assays. If they are impacting enhancers of promoter, we can check in the lab. Yep. I don't know if it's in this line or the question.
Sorry, where did that?
That was in the line of validating at the functional level or?
I think so. The question is from Dominique Rocha.
Ah, okay, yes, it's at functional level, yes. Okay. But yes, we are interested to
validate.
To validate some of these mutations.
Yep. Okay, thank you. I think these were the... Dominique replied at Maria, yes, it was functional validation. Okay. Question from the audience here, or comments? Have a?
I have a question for Andreas. In the chicken or in avian species, you have a lot of microchromosomes that are not well covered by the 50K from what I saw. Would it be more interesting first to improve the coverage of these microchromosomes?
Yes, that's absolutely right. When we built the 50K, the annotation was available back then, didn't cover the microchromosomes. So you're absolutely right. The problem is that because the 50K doesn't cover the microchromosomes, I cannot impute them. But if we update the 50K to have SNPs in the microchromosomes, you're absolutely right. Those microchromosomes, they are gene-rich, they are more important. So, I mean, again, for the benefit of audience, the chicken genome is 1 billion bases long. The first chromosome is 200 million bases, so it's 20% of the genome. Those microchromosomes are 3 million, so they are tiny. People tend to neglect them. But yeah, I absolutely agree that because as of course you point out, we only accounted for 28 chromosomes. And the other omission was that we didn't include chromosome 16. Chromosome 16 harbors the MHC complex, which obviously is important. But then again, because of due to the repetitive nature of the chromosome 16, the 50K was not doing well because again we didn't have improved annotation. So these are things that need to be fixed and could potentially have an impact. Yeah.
Just a comment on the chromosome 16. It's 90% repeat, and the genes are just synced in middle of the repeats, so finding SNP in this kind of region of this kind of chromosome, it's going to be very challenging. Honestly, I don't think it's even possible to improve it in the chromosome like the 16 of the chicken.
Okay. So do you think that, for instance, low-pass sequencing, so because that's the other option, right? Low-pass sequencing becomes more and more affordable as an option. So we don't use SNP panels anymore. But in a low-pass sequencing, do you think that it'll be easier to
Find SNP?
Yeah, or not find an SNP, but basically impute up to the whole genome, right?
Yeah, I suppose so. But you still have this challenge of this tricky chromosome. Yeah, as I said, I discovered the chromosome 16 because I worked on the MHC, and I've been astonished by the composition of this chromosome. So yeah, the microchromosome of the chicken is going to be still very challenging with any technologies, long reads and even short reads.
Okay. Other questions? Actually, I had a question for Carl-Johan. I hope he's still online. So I think that the results show very nicely the enrichment of the 42 regions that you identified in your QTL and your GWAS analysis. But did also the function annotations help you to further fine map these regions, or how could that be utilized?
I'm not sure how that could be utilized, actually, because I think in order to do that, we would need a validation set which is valid. Maybe the selective pressures, especially because you have to remember that these chicken have been selected on one trait and one trait only, which means that the deleterious effects have just been ignored. As long as they weigh a lot, they've just been propagated generation by generation. So there is definitely a chance that we will find an overlap between these peaks and broiler GWAS regions. But I do think that many of the things that have been selected in these lines, they're not tolerated in the broiler industry, because they come with deleterious side effects. For example, the high-line individuals, they get metabolic disease, and they have to be feed restricted, or else they become really ill. So what I'm getting at is that I don't think we can use our data to check if we can do fine mapping using this data, but I think we should check the intersections with QTL and GWAS peaks from other sources of fast-growing chicken.
Okay, thank you. Final chance? No one? Then I think then we can stop here. Then thank you all for your contributions and participation, and I think we break for lunch, for a networking lunch, and then we reconvene at 1:45. Quarter past 2, sorry. Yep, okay, thank you.